Mesdames, Messieurs, bonsoir. Bonsoir et bienvenue à cette première conférence du Colloque Wright pour la science, dont c'est la 17e édition cette année. Mon nom est Olivier Dessibourg, je suis responsable de la rubrique Science au quotidien Le Temps, à Lausanne. On était à Genève, on a déménagé, on est encore entre les deux villes. Comme chaque année, nous avons donc le privilège, le grand privilège, le plaisir aussi d'accueillir des orateurs de renom ici à l'Université de Genève pour ce Colloque Wright, des orateurs qui viennent du monde entier. Cette année ne fera bien sûr pas exception, puisqu'on a même le plaisir et l'honneur d'accueillir un prix Nobel, ou plutôt une prix Nobel, puisqu'il s'agit de Linda Buck, qui est l'une des rares femmes à avoir obtenu le prix Nobel de médecine et de physiologie. C'était en 2004. Elle va bien sûr s'exprimer sur le thème de cette année, qui a été choisi pour cette année, écrit ici, décoder le livre de la vie, la révolution génomique. Cette soirée se déroulera en deux parties. Il y aura bien sûr d'abord la conférence, qui sera en anglais, mais vous avez tous dû recevoir des casques de traduction simultanée de l'anglais vers le français. Il y aura ensuite une partie de questions-réponses avec non seulement Linda Buck, mais aussi à des autres orateurs de ce colloque, à savoir Michael Snyder, qui va nous rejoindre sur scène tout à l'heure. Cette discussion se fera, elle, en anglais, mais vous pourrez bien sûr poser vos questions en français ou en anglais grâce justement à la traduction simultanée. Je vous demande maintenant de bien vouloir mettre vos téléphones sur silencieux, de ne pas les éteindre, et vous comprendrez pourquoi tout à l'heure, de ne pas les éteindre, mais de les mettre sur silencieux. Avant d'entrer dans le vif du sujet, j'aimerais donner la parole à deux personnalités, qui vont nous donner quelques mots de bienvenue. Et la première d'entre elles est le recteur de l'Université de Genève, Yves Flukiger, que j'invite sur scène.
Monsieur le Président de la Fondation Wright, Mesdames et Messieurs les membres de la Fondation Wright, Mesdames et Messieurs les membres de la famille Wright, c'est pour moi un immense privilège de vous accueillir ce soir dans cet auditoire pour cette semaine exceptionnelle qui nous permettra d'entendre des sommités internationales mondiales dans leur domaine de spécialisation. C'est un cadeau que nous offre toutes les deux ans, la Fondation Wright. Et cette année, encore une fois, nous aurons le privilège d'entendre des personnalités scientifiques de très haute réputation dans leur domaine de spécialisation. Les relations entre la Fondation Wright et l'Université de Genève sont des relations à la fois anciennes, très anciennes, et extrêmement riches et fructueuses. Elles datent effectivement de 1984 et on célèbre cette année la 17e édition de ces colloques Wright qui permettent chaque année d'accueillir des personnalités scientifiques et surtout d'établir cette relation que nous souhaitons entretenir entre le public, la science et les milieux académiques. C'est le but de la Fondation Wright et c'est notre objectif également comme université de pouvoir diffuser les progrès de la science, les mettre en débat comme nous aurons l'occasion encore de le faire cette semaine. La contribution de l'Université de Genève est en particulier de mettre à disposition sa logistique, de mettre à disposition son service de communication pour qu'on ait un public aussi nombreux que cette année, une nouvelle fois. Mais c'est aussi évidemment de pouvoir mettre à disposition de la Fondation Wright, les chercheurs qui, à l'Université de Genève, déploient leurs activités, leurs recherches dans des domaines qui font le sujet de ces différentes conférences. Et j'aimerais remercier ce soir en particulier le professeur Yvon Rodriguez qui a contribué très largement à la préparation de ce programme que nous vous offrons cette année une nouvelle fois. Les relations avec la Fondation Wright sont effectivement aussi de mettre en place toute une série d'événements qui mettent en relation le public genevois avec la science et l'académie. Et c'est en particulier dans le cadre des passerelles que nous organisons avec la Fondation Wright que nous avons l'occasion d'établir cette diffusion du savoir scientifique auprès d'un large public qui fait qu'aujourd'hui, comme les années précédentes, c'est devenu un événement incontournable de la vie médiatique, publique et scientifique de Genève et j'en suis particulièrement heureux en tant que recteur de cette université. Et puis vous l'avez vu, pour ceux et celles qui n'ont pas encore eu l'occasion de l'observer, vous aurez l'occasion peut-être à la fin de cette conférence de vous rendre près du bâtiment Unibation. Vous aurez l'occasion de voir ce son et lumière qui vous est offert aussi par la Fondation Wright, encore dans cet esprit de diffusion du savoir scientifique auprès d'un public très large. C'est effectivement le but de la Fondation, de créer de l'émotion autour de la science. Et je vous assure que ce spectacle Son et Lumière que nous offre la Fondation Wright est parfaitement dans cette perspective de la création de l'émotion autour de ces événements scientifiques. Voilà, sans plus tarder, j'aimerais passer maintenant la parole au président de la Fondation Wright, le professeur Thierry Courvoisier. Et surtout, je vous souhaite à toutes et à tous une excellente soirée ce soir, mais également une excellente suite de semaine, puisque vous aurez l'occasion d'avoir encore d'autres conférences qui vous entretenir durant cette semaine. Merci infiniment de votre présence.
Merci, Monsieur le Recteur, Madame Wright, chers amis. C'est un très grand plaisir de vous souhaiter la bienvenue à la première conférence du Colloque Wright 2016, Décoder le livre de la vie. Une science, même de tout premier plan, n'a de sens que partagée. Le savoir ne porte de fruits que mis en commun au sein de la communauté scientifique, ce sont nos publications, et mis en commun entre la communauté scientifique et la société en général. La Fondation Wright apporte sa contribution à ce deuxième aspect de la communication scientifique. Elle le fait avec enthousiasme en finançant un certain nombre de projets chaque année, en organisant ce colloque tous les 2 ans et pour la deuxième fois en mettant sur pied un spectacle qui illumine le bâtiment Unibastion pendant 3 semaines. Ce faisant, la Fondation permet à ceux qui le souhaitent, à vous, d'entendre les résultats les plus importants de notre époque de la bouche de leurs auteurs, et elle permet au plus grand nombre de vivre un moment dans une harmonie science-poésie beaucoup plus proche de la démarche scientifique que la plupart ne le pensent. Nous sommes aussi très heureux de pouvoir organiser mercredi une rencontre entre des scientifiques et les collégiens de la région. Ce sont ces collégiens qui deviendront la société de demain et pour certains, nous l'espérons, les piliers de la science de leur temps. Ce colloque ne serait pas possible sans la collaboration de l'université. J'aimerais remercier ici chaleureusement l'université, son recteur, son service de la communication, pour la très belle collaboration que nous avons avec elle. Et j'aimerais remercier beaucoup, mais vraiment beaucoup, toutes les personnes qui ont œuvré depuis des mois à la réalisation de ce colloque. Les personnes de l'université, du service de communication, Fannen Sisban en tout premier, et les personnes de la Fondation, qui sont Ascension Lozano et Yvan Rodriguez aussi, qui est l'âme scientifique de la semaine que nous allons vivre. Je me réjouis d'entendre nos orateurs de cette semaine. Je les remercie de leur disponibilité. J'espère que vous tous profiterez pleinement des 4 conférences de cette année et que vous vous laisserez porter par la beauté du spectacle au Bastion. Bonne semaine.
Merci, Monsieur Courvoisier. On l'a dit, cette année, le thème choisi pour ce colloque est « Décoder le livre de la vie, la révolution génomique ». Pourquoi ce thème? Comment est-ce qu'on en est venu à choisir ce thème-là? Pour en parler, j'invite Yvan Rodriguez, qui est professeur au département de génétique et évolution à l'Université de Genève et aussi membre du comité scientifique de la Fondation Wright.
Merci. Bonjour à tous, soyez les bienvenus. Dans le but d'alléger ma courte allocution, je vais la débuter lâchement en m'associant simplement aux introductions protocolaires de mes collègues. Il est des révolutions, et j'entends par révolution des changements de paradigme ou alors des avancées technologiques majeures qui ont transformé nos sociétés. Alors je pense par exemple à la découverte des antibiotiques, je pense à l'invention de la roue, je pense à la révolution copernicienne, par exemple. Ces révolutions sont rares, mais si on a un peu de chance, si on est au bon moment, si on est au bon endroit, eh bien, on peut les vivre. Et il se trouve qu'aujourd'hui, justement, nous sommes tous ici témoins d'une révolution en marche. Cette révolution, celle qui fait l'objet de ce colloque, on l'appelle révolution génomique. C'est une révolution parce que le mystère du substrat de l'information de la vie, de l'information qui est nécessaire à la construction et au fonctionnement du vivant, c'est-à-dire une question primordiale s'il en est, eh bien ce mystère a été levé. Nous savons que cette information, ce livre, le génome, est codé dans une longue chaîne d'ADN. Conceptuellement, ce que je vous mentionne ici, eh bien on le sait depuis 50 ans. Mais pratiquement, la réalité est qu'on est capable de séquencer le génome dans vertébrés ou dans mammifères depuis l'an 2000, un tout petit peu après. Depuis donc le début de ce nouveau millénaire, qui a vu d'abord les génomes de la souris et de l'humain entièrement séquencés, eh bien nous avons décrypté le livre de centaines d'autres espèces, comme par exemple l'érable, la girafe, et pour être de saison, la chanterelle. Et on a eu beaucoup de surprises. Cet accès au contenu de l'information qui siège à l'intérieur des génomes, sans en avoir l'air, change beaucoup de choses. Il y aura en avant et en après la révolution génomique. Dans 1000 ans, dans 10 000 ans, dans 100 000 ans, en pensant à l'histoire de l'humanité, on ne pensera pas à l'iPhone, on ne pensera probablement même pas au moteur à explosion. Par contre, on pensera à la première fois où l'homme aura ouvert et feuilleté les pages du livre des espèces qui l'entourent et du livre, évidemment, de sa propre espèce. Et c'est pour vous parler de ce que l'exploration des génomes est en train de changer ou a déjà changé que s'inscrit ce colloque Wright 2016. Nous aurons, comme il vous a été mentionné, 4 conférences, chacune d'entre elles traitant dans domaines différents, mais toutes très directement affectées par les problèmes et les progrès de la génomique. Aujourd'hui, nous ouvrons le colloque avec le professeur Buck, Linda Buck, qui nous parlera de sa découverte de la plus grande famille de gènes présente dans les génomes de vertébrés. Avec elle, nous allons donc parler de l'identification de gènes au service de la compréhension des mécanismes de base de la biologie. Demain, nous écouterons le professeur Snyder, qui est ici présent et qui dirige le Centre de génomique et de médecine personnalisée de Stanford. Il nous parlera de ce que la caractérisation génomique individuelle chez l'humain va changer dans notre compréhension des maladies et dans notre façon de traiter ces maladies. Puis jeudi, Svante Pääbo nous parlera d'évolution. Il nous parlera de notre histoire, à nous les hominidés, et en particulier du séquençage du génome d'une espèce cousine, l'homme de Néandertal, et de ce que ce séquençage nous dit sur notre propre évolution, sur notre propre espèce. Et finalement, nous clôturerons ce colloque vendredi avec une conférence du philosophe Peter Sloterdijk qui nous fera partager sa réflexion sur le futur de notre espèce. Alors pourquoi est-ce que la révolution génomique affecte cette réflexion ? Eh bien simplement parce que non seulement nous pouvons aujourd'hui séquencer la totalité du génome d'un individu, mais nous pouvons également le modifier. L'idée est donc ici, lors de ces 4 conférences, de transmettre non seulement la connaissance scientifique pour nous grandir, pour nous élever, pour nous rendre plus conscients de ce qui nous entoure, et peut-être aussi, comme le suggérait mon collègue précédemment, faire naître quelques vocations parmi les plus jeunes d'entre nous, mais aussi d'accroître notre capacité à discerner le vrai du faux et de fournir un substrat solide et structuré à une réflexion. Et ceci est particulièrement critique dans notre beau pays, qui, contrairement à ses voisins, fait souvent confiance à son peuple lorsqu'il s'agit de prendre des décisions à propos de sujets relativement pointus. Et donc voilà pour le choix de la thématique de ce colloque pour cette année, et ainsi également pour le rôle que nous espérons, à la fois la Fondation Wright et l'Université de Genève jouer au sein de la Cité.
Voilà, restez avec moi, Yvan, quelques secondes. Je vous parlais de ne pas éteindre vos téléphones portables, de les mettre sur silencieux. Pourquoi? Comme vous le savez, comme je l'ai dit tout à l'heure, il y aura une partie de questions-réponses à la suite de la conférence de Linda Buck. Il y aura les traditionnels micros qui circuleront dans la salle. On vous encourage évidemment à poser des questions, en français ou en anglais à nouveau. Et cette année, on va tester une nouvelle manière de fonctionner aussi, puisqu'on va vous donner la possibilité, à vous, mais aussi à ceux qui nous regardent via le live stream Internet, qui ne sont pas là ce soir, de poser des questions à travers Twitter. Donc pour cela, on vous invite à vous connecter sur le réseau Wi-Fi de l'Université de Genève, c'est Guest Unige, et à utiliser Twitter en mentionnant le hashtag, donc le dièse, Colloque Wright. Posez vos questions à travers ce biais-là, et Yvan va les trier et va les relayer aussi durant la discussion à nos deux panélistes. On vous encourage vraiment à le faire. Posez des questions là que vous n'oseriez peut-être pas poser avec le micro à la main. C'est l'occasion vraiment de le faire. Voilà, maintenant, mesdames et messieurs, place à notre conférencière de ce soir.
Merci. Alors, je vais faire très court. Nous avons donc aujourd'hui le plaisir et l'honneur d'accueillir Linda Buck. Pour mentionner très brièvement son parcours, Linda Buck, après avoir reçu un doctorat en immunologie de l'Université du Texas, rejoint pour un stage postdoctoral le laboratoire de Richard Axel à l'Université Columbia à New York. C'est là qu'elle va identifier les premiers membres de la superfamille des gènes codant pour les récepteurs à odorants. Elle établit ensuite son propre laboratoire à Harvard, puis migre sur la côte ouest des États-Unis et à Seattle, où elle poursuit aujourd'hui ses recherches. Son parcours tout à fait remarquable a été jalonné de découvertes importantes et nombreuses, et de récompenses prestigieuses, trop nombreuses pour les citer ici. Mais je vais tout de même mentionner évidemment le prix Nobel, qui a déjà été mentionné précédemment, qui lui a été attribué en 2004, justement pour sa découverte des gènes codant pour les récepteurs odorants. Je vais donc laisser la parole à Linda Buck, qui va nous faire voyager dans un système sensoriel présent chez tous les vertébrés, un système qui nous permet d'extraire de l'information du monde extérieur, du monde chimique qui nous entoure, qui nous permet de transformer ce monde chimique en percepte qu'on appelle une odeur. Dear Linda, welcome in Geneva. We're glad and honored to have you here. The floor is yours.
Thank you so much, Yvonne, for that lovely introduction. So it's really an honor for me to be here, to be part of this week's H.W. Wright 2016 colloquium, the topic of which is the genomic revolution. I'd like to thank the Wright Foundation and Thierry Courvoisier, its president, and the scientific organizer of this colloquium, Yvonne Rodriguez, for inviting me to participate this week and for putting together a wonderful program of talks and events. And I hope that you all get a chance to see the Sound and Light show, which we saw last night and which was absolutely spectacular. I'd also like to take the opportunity to thank Olivier Desiborg for his part in the colloquium and for his very important role as a journalist in bringing science to the public, which is, of course, the goal of the Wright Foundation's colloquia and many of their other activities in Switzerland. I believe the genomic revolution is a very timely and important topic as a colloquium topic. In recent years, we've seen an explosion of new genomic technologies. And we have seen as you've already heard, the sequencing of the entire genomes, that is all the genes in humans and in many other species. We've seen the identification of functions for specific genes. And we've seen the identification of genes involved in many different diseases. In the field of neuroscience, which is my field of study, the genomic revolution has provided new tools with which to begin to understand how the brain generates thoughts, perceptions, and emotions. These are still mysteries, but the tools that have been developed by the genomic revolution are proving extremely important in deciphering how the brain works. Today, I'd like to discuss how we've used some of these genomic tools to try to understand the sense of smell, one of the 5 senses through which we perceive the world around us. Through the sense of smell, humans and other mammals can perceive a vast array of chemicals in the external world as having a distinct odor. All of these so-called odorants are small volatile molecules, but they have different structures, and somehow those different structures are perceived as having different odors. The olfactory system is the system that governs the sense of smell, and this system is characterized by exquisite sensitivity and discriminatory power. Even the slight change in the structure of an odorant molecule can change the way it smells to us. For example, these 2 molecules have similar structures, and yet one smells like pear and the other one like banana. Now, in addition to these odorants that are perceived as odors, the olfactory system in animals also detects what are considered social cues. Some of these social cues are pheromones. Pheromones are chemicals that are released from animals that act on members of the same species to stimulate hormonal changes or innate behaviors, such as mating or aggression. The olfactory system of animals also detects predator odors, which can elicit an instinctive fear response. Now, over the years, we've been especially interested in 2 questions. First, how do mammals detect so many different chemicals, so many different odorants? And second, how does the nervous system translate these different chemicals into diverse odor perceptions and innate responses? Now, mice have 2 olfactory pathways. The main olfactory pathway, which is also found in humans, and the vomeronasal pathway, which is found in many animals but not in humans. in the main olfactory pathway, which can detect odorants as well as some social cues. These chemicals are detected by neurons in the olfactory epithelium, which lines the nasal cavity. These neurons transmit odor signals to the olfactory bulb of the brain, which relays those signals to the olfactory cortex. From there, the odor signals travel to higher cortical areas, which are thought to be important to odor perception and odor discrimination, as well as to deep areas of the brain that control emotions and basic drives such as mating and parental behaviors. In contrast, the vomeronasal organ, or VNO, in the vomeronasal pathway detects primarily social cues. And information from neurons in that organ travel through the accessory olfactory bulb to the vomeronasal amygdala and then on to deep brain areas, again involved in basic drives and emotions. The olfactory epithelium of the nose contains millions of olfactory sensory neurons. Each of these neurons extends a number of rope-like structures called cilia into the layer of mucus that lines the nasal cavity. These cilia is where the neuron detects odorants that are dissolved in the nasal mucus. In our initial studies, we asked how it is that these cilia can detect odorants. What is the molecular basis of this detection? And in those studies, Richard Axel and I discovered the odorant receptor family. This family is the largest known family of genes. Mice have about 1,000 different odorant receptors, or ORs, and humans have about 350. This is about 1 to 5% of all the genes in the genome. Seen here is a schematic of an odorant receptor, OR, in the membrane. It has 7 transmembrane domains. It loops back and forth through the membrane 7 times. These red balls, the balls indicate amino acids that make up the protein. The red ones are ones that are especially variable from one OR to the next. And this hypervariability in the amino acid sequence of these proteins is what allows different ORs to detect different odorants. This slide shows a schematic of an OR in the plane of the membrane. And here are the odorant molecules. It's thought that an odorant molecule will enter a pocket in the plane of the membrane formed by a combination of these transmembrane domains. And when it does that, it stimulates the receptor, and that in turn activates the neuron, so the neuron transmits signals to the olfactory bulb of the brain. With the sequencing of the human genome in about 2000, we and others asked how the OR genes are organized in the genome, on the chromosomes. And surprisingly, these studies showed that OR genes can be found on most human chromosomes. The same thing was found in the mouse. So oftentimes there are clusters of OR genes. As you can see here, the green ones are intact genes. Many in humans are actually pseudogenes. They're non-functional. Those are in red. Now, as with other genes, OR genes are composed of a stretch of DNA. And when the OR gene is, quote, expressed, an RNA copy is made of that gene, and that in turn is translated into the protein. Following the discovery of the odorant receptor family, we and others found a number of other families of chemosensory receptors in the olfactory system. This included a very small family of tar receptors in the nose and 3 families of receptors in the vomeronasal organ, one of which, the FPRs, was identified by Ivan Rodriguez here in Geneva. Now, the discovery of the odorant receptors, which is my main topic tonight, it did two important things. First, it told us how it is that we can detect odorants in the nose. And secondly, it provided a set of molecular and genetic tools that could be used to explore how the nervous system translates odorant chemicals into odor perceptions. So one of the first questions that we and others asked after the discovery of the receptors was, how is it that signals from 1,000 different receptors are organized in the nose and in the brain? And in the nose, as I mentioned, there are millions of olfactory sensory neurons in the mouse, and that's true in humans as well. Each of these neurons sends a single axon, a long extension to the olfactory bulb of the brain. There, the axon enters a spherical structure called the glomerulus, and this is where it connects with the bulb neurons so that it can talk to the bulb neurons, transmit signals to the bulb neurons, which they in turn send on to the olfactory cortex. Now, in early studies, we and others used OR gene probes to look at OR messenger RNA in slices through the olfactory epithelium. And what these studies showed was that the olfactory epithelium of the nose has a series of spatial zones that express different sets of OR genes. Here you can see zone 1, 2, 3, and 4, for example. Each OR gene is expressed in a very small percentage of the olfactory neurons, and those neurons are scattered randomly throughout one of these zones. When we looked at genes expressed in single neurons, we found that each olfactory sensory neuron, or OSN, is likely to express only one of the 1,000 different OR genes. So these studies told us two important things. First, information from a given OR is highly distributed in the nose. So this indicated that neurons with receptors for a particular odorant, like strawberry, must be interspersed with neurons with receptors for other odorants, such as lemon. The second thing it told us was that inputs from different ORs are segregated in the nose. So that means that each neuron in the nose sends signals to the brain that reflect the odorant specificity of one kind of OR. Now, when we looked at the olfactory bulb of the brain, we found something totally different. Here, we found that the axons of those neurons that are scattered in the nose and have the same receptor all converge in a few glomeruli at two spots, one on either side of the bulb. And surprisingly, those glomeruli had similar locations in different individuals. And it's still not understood how that happens. But Ivan Rodriguez and some other olfactory researchers have hints as to what might be going on that allow those axons to converge at the correct place in the olfactory bulb. So together, these studies indicated that the OR family is first, information from the family is highly distributed in the nose, in the olfactory epithelium, but then in the olfactory bulb is transformed into a semi-stereotype sensory map, which is similar in different individuals. Note, though, that there's one important thing that's similar between the nose and the olfactory bulb. At both locations, inputs from different ORs are segregated. So each neuron in the nose and each glomerulus in the bulb appears to be dedicated to only one OR. In the olfactory cortex, the next level of the olfactory pathway, it seems as if things change again. It's not clear exactly how OR inputs are organized, but it looks like they become highly distributed again. So another important question early on was, how does the OR family generate a multitude of different odor perceptions? We found that when we tested different but related odorants, that a single OR could recognize multiple odorants. And one odorant could be recognized by multiple different ORs. But interestingly, different odorants were recognized by different combinations of ORs. So this indicated, excuse me, that the OR family is used in a combinatorial manner to encode odor identities. Different odorants are recognized by different combinations of receptors and thereby encoded by different combinations. But each OR can serve as one component of the codes for many different odorants. So this is somewhat related to the way in which different letters of the alphabet can be used to form a multitude of different words. Now, these studies also provided insight into a few puzzling features of human odor perception. As I mentioned earlier, changing the structure of an odorant can sometimes change the way it smells. And we found that when we looked at odorants that have similar structures but different perceived odors in humans, such as rancid, sour, and goat-like versus sweet and woody, in every single case, the different structures but related structures were recognized by different combinations of ORs. So if you change the structure of an odorant even slightly, you can change its combinatorial code, and this can lead to a change in perceived odor. Now, even if each odor were recognized by only 3 receptors, this combinatorial coding scheme could generate, in theory anyway, millions of different odor codes. And this, in turn, can explain how we can not only detect a multitude of odorants, but how we can distinguish among different odorants, even if they're almost identical in structure. We also found that changing the concentration of an odorant can change its combinatorial receptor code. With increases in concentration, we saw the recruitment of additional ORs into the odor response. This may explain why it is that odorants sometimes can have different perceived odors at different concentrations. So altogether, these early studies indicated that the spatial code for an odorant in the nose is a dispersed ensemble of neurons, each expressing one component of the odorant's receptor code, whereas in the bulb, it is a specific combination of glomeruli that receive input from those ORs and have a similar arrangement in different individuals. So I'd now like to turn to more recent work that we've been doing where we've made use of some very new technologies. In one case, a new genomic technology. In the other case, one that's related that uses viruses to chart neural circuits in the brain. Excuse me. So the organization that we and others discovered in the nose and the olfactory bulb raised a number of intriguing questions about how the structure of the olfactory system is created during development. One question is, how does the olfactory sensory neuron, the OSN, come to express only 1 out of 1,000 OR genes? How could it do that? Now, the olfactory sensory neurons, or OSNs, are short-lived cells. They generally live only for a couple of months. And they're continuously replaced from an underlying layer of stem cells. This is the nasal lumen, the surface of the epithelium up here. And we have progenitors giving rise to neuronal precursors, which in turn give rise to immature OSNs. And those immature OSNs then turn into mature OSNs. And the genes expressed by these neurons change from cell type to cell type, although it wasn't known but a few what genes they actually expressed. But there were known some so-called marker genes that you could use to tell what type of neuron it was. For example, mature neurons express OMP. So to investigate when and how the neuron activates an OR gene for expression, Naresh Hanchade in our lab, a postdoc, decided to use a new technology called single-cell RNA sequencing, or RNA-seq. So this technology allows you to look at all the genes expressed in a single cell. So what he did was to take the olfactory epithelium of the mouse, dissociate the cells into single cells, put one cell per tube, and then make a DNA copy, complementary DNA copy, or cDNA copy of each RNA in the cell, which was confined to the tube. He then amplified those DNAs and to make a single cell cDNA library from each tube, sequenced those libraries, and that identified the genes expressed in each neuron, which is called its transcriptome, because it's transcribed, the gene is transcribed into RNA. So we then collaborated with Cole Trapnell, who's a computational biologist, and compared the transcriptomes of different cells in each of these 4 classes that I mentioned. And this comparison showed a linear trajectory from progenitors through the mature OSNs. Interestingly, we found waves of changes in expressed genes during development, going from the progenitors through the precursors to the immature neurons to the mature neurons. And as you can see in red here, or in lighter colors, we can see these waves of clusters of genes changing in waves across development. And there was a total of almost 4,000 genes that were differentially expressed among the 85 neurons that we looked at in these studies. So these waves, these clusters of genes, could conceivably contain genes that are important to these changes of one neuron type into the other, and possibly the selection or activation of individual OR genes. So in terms of the ORs, we first saw OR RNAs appearing at the precursor to early immature neuron stage. And the number of RNAs per cell encoding the ORs increased gradually and unevenly into the mature neuron stage. We found that most mature neurons express a high level of a single OR, although some also expressed a second OR, and even a third in one case. However, we saw something very different in the immature neurons. To our surprise, we found low levels of multiple OR genes expressed in the immature neuron stage. In fact, Naresh found up to 12 different OR genes expressed in a single neuron at this early immature stage. Now, of course, we were worried that maybe it was an artifact of some kind. So, I said, okay, no validation in the nose, no publication. So it took a long time to detect these very low levels of RNAs for ORs in the nasal neurons, but eventually Naresh was able to do it. And again, he could validate the expression of more than one OR gene per cell in a slice through the nose using differently colored probes, fluorescent probes, recognizing different OR messenger RNAs, as you can see here. These he mostly saw in the neonatal animal, where there are many immature neurons, and very few in the adult animal, where most of the neurons are mature. So as I mentioned earlier, the OR genes are found on most chromosomes. That's true in human and in mouse, and they're often clustered. So one thing we thought was, well, how do you get these multiple OR genes expressed in a single neuron? One thing we thought of was that maybe a single location was activated on a particular chromosome, and so initially it could express low amounts of multiple OR genes at the same locus. But what we found was that a single neuron could contain genes located on up to 7 different chromosomes. So somehow the neuron is able to turn on genes that are scattered throughout the genome. However, using, again, the 2 colored probes for different ORs, we found that genes, OR genes expressed in the same neuron were invariably expressed in the same zone in the nose or in partially overlapping zones. So this suggested that when the immature neuron initially activates multiple OR genes, those are restricted to a particular zonal subset of OR genes. And then with time, as the neuron matures, it can come to express only one of those OR genes in the mature state. We have no idea how this happens, but we're investigating that now. We'd like to know the mechanisms used to initially activate the genes and the mechanisms that must be operating to narrow the number of genes to only one and to get a high-level expression in the mature neuron. Okay, so now I'd like to switch to a completely different topic. And I mentioned earlier that in animals, social cues can cause innate instinctive behaviors and hormonal changes. So how do pheromones control sex hormones? How do predator odors induce fear? How do some odorants elicit feelings of pleasure? That's true in humans as well as animals. And how do some smells stimulate appetite, even when we don't want them to? So what are the neural circuits that control these responses? Now, mice and many other animals show instinctive fear responses to predator odors. They show particular fear behaviors. They either try to escape, or if they can't escape, they become immobile, a behavior that's called freezing. And they also show increases in blood levels of stress hormones. And interestingly, humans show similar responses to fear and stress, as you can see illustrated here on the right. So Kunio Kondo, another postdoc in the lab, wanted to explore how it is that predator odors can elicit stress hormone responses. And he decided to focus on a very small subset of neurons in the hypothalamus of the brain called corticotropin-releasing hormone, or CRH neurons. And you can see them clustered here bilaterally in this small area of the hypothalamus. These neurons control stress hormone increases in response to predator odors, as well as in response to other forms of fear and stress. And they're also implicated in fear and stress in humans as well as in mice. So we hope that by understanding these stress and fear pathways, not to predator odors, because most of us are not afraid of predators, at least we don't encounter them. Although I must say that our neighbors sent us an email the other day saying that there was actually a bear in our neighborhood, which I have never heard of before, so I'm a little bit wary now of going outside. But anyway, usually we don't have to fear predators because we don't really encounter them. So to study the neural circuits underlying fear responses to predator odors, and also to study other neural circuits, we're using Cre-dependent pseudo-rabies viruses, or PRVs, in order to chart the neural circuits. These are not related to rabies viruses, actually. They're actually herpes viruses. So I won't go into the details of why these are dependent upon the enzyme Cre recombinase, but I will say that if one of these Cre-dependent PRVs infects a neuron that has been engineered to express Cre, the virus will replicate in that neuron and then travel to upstream neurons. So it infects and thereby labels a chain of connected neurons that transmit signals to the downstream Cre-expressing neuron. So the idea is shown here, if you're interested in olfactory circuits. Here we inject a Cre-dependent PRV into the vicinity of Cre-expressing CRH neurons. They replicate in those neurons. The PRVs then travel to the olfactory cortex or vomeronasal amygdala, then to the olfactory bulb, and then, if we're lucky, to the olfactory epithelium and or VNO. So Cuneo was interested in the higher olfactory areas, and he wanted to know if there was a specific area of the olfactory cortex or vomeronasal amygdala that's responsible for the stress hormone responses to predators. Now, this was because the olfactory cortex, which receives input from the nose, is actually very complex and it's not understood. It has about 11 distinct areas, and basically it's not known what they do, those different areas do. There are some hints about what a few of them do, but for the most part it's unknown. And the vomeronasal amygdala also contains 2 different areas. So what Cuneo did was to use Cre-dependent PRVs, inject them into the vicinity of CRH neurons that express Cre, and then he waited for different periods of time, and he made slices through the brain and looked to see where the virus had gone to after infecting the CRH neurons. And what he found was the virus traveled to a number of different brain areas, indicating that the CRH neurons receive input from neurons in many other areas of the brain. And at least 31 of those areas are directly upstream. They're sending signals directly to the CRH neurons. And then others can be indirectly connected. So among these areas were parts of the olfactory cortex and the vomeronasal amygdala. So rather surprisingly, Cuneo found PRV infected neurons in 5 of the 11 areas of the olfactory cortex and in both areas of the vomeronasal amygdala. So this suggested that, well, we didn't know what to think actually, but it raised the possibility that these different areas are all sending the same signals about predators to the CRH neurons. So to investigate that possibility, Cuneo exposed mice to 2 different predator odors that are detected in the nose, bobcat urine, and TMT, which is a fox predator odor. And then he asked, where were the PRV neurons in the higher olfactory areas that were activated by these predator odors? And what he found was that there was only one small area of the olfactory cortex that showed a significant activation of PRV-positive neurons. This was the ampere, or amygdalo-piriform transition area. And we were surprised in part because we had never even heard of it. I mean, it was there in the brain atlas, but we were, it's a very small area, and so we thought, well, that's interesting, but, you know, is it real? I mean, is that area really involved in these predator odor responses? So to investigate that question, Cuneo took what's called a chemogenetic approach. Okay, so in this approach, he used what are called DREADs, designer receptors exclusively activated by designer drugs. And the designer drug here is a synthetic drug called CNO, and the receptors are mutated receptors that can be activated by CNO, and they cause either neuronal activation or neuronal silencing. So Cuneo injected a virus, not a PRV, another kind of a virus, encoding either the activating DREAD or the silencing DREAD into the ampere. So after injecting the activating DREAD, he gave the animal CNO. And what happened was that he saw a huge surge in blood stress hormone. This was ACTH, adrenocorticotropic hormone. So, and also he saw the activation of CRH neurons. So this indicated that indeed neurons in ampere could activate the CRH neurons, causing an increase in blood stress hormone, which mimicked an instinctive fear response. Conversely, when he silenced ampere with the silencing DREAD, he found that the synthetic CNO chemical caused a striking decrease in the ability of predator odors to cause a stress hormone response. So together these studies indicated that the ampere plays an important role in the stress hormone responses to predator odors. And what we'd like to know now is what the other neurons are doing, that we saw the other PRV-infected neurons in the other olfactory cortical areas. But that remains to be seen. But I will mention something in a minute with regard to that. Interestingly, as I mentioned earlier, the predator odors cause not only a stress hormone increase, but also particular behaviors. And one of them, as I mentioned, is freezing behavior. So if an animal is confined to a cage and it can't escape, and you expose it to a predator odor, it will just sit in the corner and not move. You know, presumably thinking that if it doesn't move, the predator won't know it's there. So what Cuneo found was that in contrast to the stress hormone response, where silencing ampere inhibited the response, the silencing ampere had basically no effect at all on the freezing behavior. So that indicates that while the ampere is important in the stress hormone response, the other component of the fear, instinctive fear response, the behavior, does not involve ampere at all. So we'd now like to know what other part of the brain is involved in that behavioral response, and studies from another lab have linked another small part of the olfactory cortex to freezing behavior to one of these predator odors. Now, as I mentioned a minute ago, we're interested in what the PRV, the neurons upstream of CRH, neurons in other areas of the olfactory cortex are doing, and also in the vomeronasal amygdala, although we have a better idea there. But interestingly, we found, consistent with some other reported studies, that some odorants, which the mice find attractive, can actually block the stress hormone response to a predator odor. In fact, we've published that the TMT, which also causes avoidance, the fox predator odor. So the animal will stay on one side of the cage and it won't go near a piece of filter paper with TMT on it. And certain attractive odorants can block that behavior. So the mouse goes up and it might even pick up the filter paper and carry it around the cage, maybe take a bite or two out of it. And we also found that a couple of different attractive odorants that the animal likes can block the stress hormone response to the predator odor. So a postdoc in the lab, Eun-Jung Lee, is looking in the brain to see how that works. How can you block these responses? Are there other stress responses being blocked? And of course, this may cause you to think of something that we have all heard of, and that's aromatherapy and spa treatments. And I do think that there may be something to those effects of odors on humans, and maybe at some point in the future, we can begin to study those, although I don't think we can get a federal grant to study aromatherapy, at least not yet. So finally, I'd like to acknowledge the very talented students and postdocs that did the work that I've discussed today, as well as a lot of other work. And I'd also like to acknowledge our collaborators at LERC in Japan, the University of Washington, University of California, Berkeley, others at Fred Hutchinson, where we're located, and Harvard Medical School, our collaborators there who gave us their CRH Cree mice. A number of these previous students and postdocs and others have gone on to establish their own labs and have trained their own students and postdocs, and that has led to an expansion of our understanding of the olfactory system and how the brain functions. So with that, thank you very much. It's been a pleasure to be here and to talk with you.
Thank you. Thank you, Lindenbach, for this fascinating presentation. Please take a seat. So, as you see, I'll switch to English, but je vous encourage tous à utiliser vos casques de traduction. Stay with us, Linda. Stay on this. Oh, sorry. Oh, yours. Yes. As I told you, we are happy tonight to have a second panelist on stage. He will be talking tomorrow, and that is Michael Snyder. Michael Snyder is a professor of genomics at the University of Stanford. Please come on stage. Welcome, Michael Snyder. Yes. So maybe first question to you, Michael. I'm sure many people here will come and hear you tomorrow. But in a few minutes, in 3 minutes, can you present what you do? What is your research? What is your daily basis work?
Sure. Are we on? Hello?
Yes.
Hello? Is it on?
Yes.
Oh, now it is, yeah. Great. So I'm at Stanford University, and I'm a geneticist. We've invented many technologies for characterizing the human genomes and the many molecules that are ultimately made from the human genome. We also these days analyze human disease. That is to say, we sequence people who have cancer to try and figure out what drugs they should be taking. We also find people who have mysterious diseases and we don't know the cause. So we actually sequence their DNA to try and understand the genetic basis of their disease. And lately, we've been sequencing lots of healthy people to try and predict what diseases they might get and we make actually billions of other measurements as well. So tomorrow I'll talk about all this kind of research, and if you're interested in how genomics affects human disease and how it might be useful for you in the future, that's what I'll talk about.
Thank you, Michael. Okay. Let me turn back to you, Linda. In the last slides, latest research you published in Nature, right? You kind of partly respond to the question, is the sense of smell innate or acquired, right? Mm-hmm. Is that correct? Can you answer that? Can we answer that question?
Well, certainly in animals, the responses to certain odorants, like social cues, are instinctive. In humans, it's not known. My guess is that we do have innate responses. You know, why else would people have similar responses to flowers, for example, or fruits versus skunk, or putrid odors? And in fact, one of the tar receptors, they're also present in humans, and we know that one of those tar receptors in humans recognizes rotten fish. Not fresh fish, but rotten fish. And most people find that to be revolting and will avoid rotten food. So I think that there is some innate aspect of the human.
You mentioned those 350, right? 350 genes. That's a huge amount of genes just for the sense of smell. But out of those 350, 50 vary between each individual, right? Which means that we're not smelling the same thing or in the same way. Is that correct?
Well, I think that's probably true, but it's not really worked out because it's very difficult to analyze what those individual receptors recognize. And there are a few cases where people have shown that there's a change, and that leads to a perceptual change. But it's not. In theory, yes, you do see changes in the receptors if you look from person to person, but it's not clear exactly what that means yet.
Yeah, we've actually done some work on this as well, believe it or not. And people do have different receptors, and there is a wide variety in perception of smells, and in fact, you may know for wine tasters, they have very different senses. It's probably relevant to a lot of people in this room, so people perceive deodorants from wine very differently. And actually people are trained, right? The sensors of tasters. Actually, some of them have, first of all, if they're insensitive, they usually are rejected as a person. So I think it'd be a lot of fun to actually match up the genetics and see what
it would
receptors.
Be fascinating.
Linda's work with people's ability to sense wine. Maybe someday in the future, your bottle of wine will be tailored to your particular odorant receptors.
Or just like personalized medicine.
Right. I'd love to see that happen.
Another question, I'm sure it happened to all of you, that you smell something and it immediately brings you to, in memories, in souvenirs that you had in the past, right? Do we know the links between the smelling system and the memory system?
No, it's not known, and people always comment on this. It's very well known that scent has this ability, unlike any other sense, to make us feel the way we felt at some time in the distant past. And it's, eventually I think we'll understand, but I don't know if it'll be within my lifetime.
What is lacking to try to understand?
Well, we don't understand the neural circuits. And we don't understand perception. We don't understand how the brain works well enough to know where those things even happen in the brain.
When I came to visit you for the interview, you mentioned those pretty new techniques that allow us to really go further. Can you tell us more about this? And maybe you can jump into the discussion because I'm sure you use them almost every day right now.
Well, there are a number of new techniques. Excuse me.
Bless you.
Sorry. That have been developed in recent years for looking in the brain, but that's mostly being used with mice, just to figure out what neurons are doing in different parts of the brain, how memory works, how learning works, how avoidance works. So that there are methods that allow you to shine a light on a part of the brain and activate neurons. There's the chemical genetics that we're using.
Michael, would you change? Yeah.
Well, I don't have a whole lot to add, but there are trillions of neurons, right? Or how many neurons are there? Linda would know, but it's very complex in terms of all the connections that are made. And that's why it's going to be a huge effort to make a map of what neurons connect to what and work out the whole pathways. So it will be a huge effort. There are very large projects being launched to try and make these maps, and it will require lots of methods. Some of the ones that Linda talked about today involve trying to look at every single cell to see what genes are turned on and off in those cells, and then actually make the maps of the connections. So it's a huge project involving probably thousands of labs till it's all done.
Will we get there one day?
I hope so. I think we'll learn some basics. I think
so, yeah.
Yeah, I mean, I think we will actually tailor specific receptors to people's behaviors at some level. Understand, at least at the genetic level, why some people respond to certain odors and others don't.
There are a few cases where a receptor has been identified in human that determines whether the person can detect the odorant or not. But that's just a few cases. And also remember that this combinatorial receptor code, when you smell a flower, it's not a single odorant. It's a combination. And that blend, the different odorants in that blend, can also modify each other's sensation. So similar to what I said about odors blocking instinctive behaviors, you can always also have odorants blocking or competing with each other at some level of the brain to modify the perception. So it's really quite complex. And there are 100 million neurons in the mouse brain. And that's already 1,000-fold smaller than the human brain. So it's a real challenge.
I think we can now open the floor to questions. Are there questions? Yes, up there.
Merci pour votre venue, professeur, de vous être déplacé dans cette petite ville de province, quoique. J'ai cru comprendre que les souris avaient un odorat plus développé que nous. Moi, ce que j'aimerais savoir, c'est combien de personnes ont été nécessaires pour établir des lignes, des règles générales qui sont devenues quasiment des lois biologiques. Deuxième question, est-ce que les industries, lesquelles vous soutiennent, dans quel but? Et la dernière, est-ce que vous avez intégré dans vos expériences des malades? Comment s'appelle cette maladie quand on ne sent plus rien? Je vous remercie. Est-ce que je résume?
On vous a compris. On laisse la traduction se faire.
Yeah, so the first question was how many labs were required to uncover those things, a combinatorial code, for example?
In the mice, yeah.
Sorry. Can you translate?
Can you ask your question again?
La question, c'est combien d'individus ont été nécessaires, ont dû être étudiés pour que vous pouviez établir ces règles et ces quelques lois? Et l'autre, c'est est-ce que vous avez des industries, dans quel but, qui vous aident aussi financièrement?
Oh, the number of mice that we used.
Yes.
I'm not sure.
So
the people working in the lab.
But
It actually wasn't very many people. The initial studies that we did on the organization in the nose, that was a student and a postdoc in my lab. And then the combinatorial receptor codes, it was one postdoc in my lab, together with our collaborators, 2 collaborators in Japan. And the olfactory bulb, that was, again, just 1 postdoc and 1 student in my lab. Industry, we don't get any funding from industry. We get funding from Howard Hughes Medical Institute, which is a nonprofit, and from the National Institutes of Health.
Est-ce qu'il y a d'autres questions? Il y a une question ici en bas, s'il vous plaît. Ou en haut.
Hello. I have a question concerning the experiment you did on the single-cell sequencing. And I was very impressed by the results and by the fact that we're able to make a difference between different cell types in such a small place in terms of gene expression. My question is, you saw that there are certain genes which are expressed in certain cells and some genes which are not expressed in certain cells. I'd like to know whether now, by keeping the mice in a different environment, you may actually also influence the overexpression of certain genes, and whether this pattern of expression could then be transmitted from generation after generation. My question is basically on the effect of the environment on the expression on those receptors in those locations.
There is some evidence that if the animal is exposed to an odorant continuously, that you can have a higher number of neurons expressing the receptor, probably because they don't die. Now, in terms of passing that on, there's also a claim that can occur, but I think that there's more study needed to establish something that unusual.
May I enlarge the question? There's this huge field of so-called epigenetics, right? We thought for a long time that everything was settled with the genes, but we discovered a few years ago that the genes are important, but not only the genes are important, the environment, right? Can you maybe elaborate on how important is epigenetics?
Sure, it turns out it's really, really important. So for example, nutrition, we know affects not only you, but also subsequent generations. There's a number of studies on that. There's some beautiful studies on exercise directly affecting the chemical modifications to your DNA. So for example, there's a group in Sweden that had people bike with one leg but not the other, and then they actually took little bits of the muscle and they showed that their DNA was chemically modified in the biking leg relative to the unbiking leg. So exercise has a direct effect on your DNA, and that's epigenetics. I'll talk about this a little bit tomorrow.
How important is that regarding to genes? I mean, is that more important, less? Do we know that?
It depends on the situations. There are some cases, your genes are important and your environment's important, and the contributions to different things is different. So your immune system is very dependent on your environmental response, but some aspects are dependent on your genetic response. I think is the microphone okay? Oh, it's my ears that are going on and off here, which is very disconcerting.
Almost like epigenetics.
Yeah, it's epigenetics.
There was a question just here in the front. La dame au troisième rang.
So thanks for this great talk. I have a super amateur question. I'm thinking a little bit about the book by Patrick Suskind called The Perfume, which I'm sure you've heard of, at least if not read, and the fascination with bad smells. And that makes me think of dogs, in particular retrievers, which will come and bring you something really disgusting. And I'm wondering, first of all, in dogs, do we know how many odor receptors they have? And why do they like these nasty smells?
Well, I don't know why they like nasty smells, but they have the number of receptors is not so different if you compare dog with mouse. But they have many more. Of course, dogs are highly sensitive to odors, and they apparently have many more neurons in the nose.
Do they have a higher density of receptors as well?
That's what I've read, but higher density and many more, like I read 300 million versus about 5 to 10 million in mouse or human.
regarding to this question, there's something, this hypothesis, right, that we kind of lost the sense of smell, or broader sense of smell, when we woke up as humans. Is that a true hypothesis or not?
Well, certainly the vomeronasal organ has disappeared and is vestigial. There's like signs of it in some people, and you can even see vomeronasal receptor genes in the genome, but almost every single one is a pseudogene. It's amazing. It's like a garbage dump of genes that aren't used anymore. Now, the human does have fewer receptors, but we're highly sensitive to odors as well. Some people say that it was with the increase maybe in color vision, the increase in the visual system, in importance and in power, allowed for the olfactory system to step back a little bit.
I'll just turn to our Twitter followers. Est-ce qu'il y a des questions du compte Twitter?
Il y en a quelques. Il n'est pas on.
C'est bon?
Oui, vous m'entendez, oui. Là, vous m'entendez. Alors, il y avait des questions sur les chiens, mais je pense là qu'on y a répondu. There are some questions about the fact that these neurons are special in the sense that when they die, they can regrow somehow. So what happens when a neuron, an olfactory neuron, dies?
Well, it just disappears. And it's not so different from other epithelia, like epithelial cells that, line the oral cavity will shed and skin cells turn over. So it's not so different from that. But the new ones come from the progenitor cells in the epithelium. So as the old ones die, new ones are born and take their place. The mystery really is how the new ones, which we think more or less randomly choose a receptor gene to express, know what glomerulus to contact, as you know. Of course, Yvonne, because you study this, how they know to go to a particular glomerulus.
Est-ce qu'il y avait d'autres questions?
Yes, there's another one which is interesting. You talked about the stress and some ORs that were involved in sensing some specific molecules that would induce stress. And the question is about how does that translate to what we know about humans sensing some molecules? What do we know about stress induced by odors in humans?
I would really that's a very interesting question, and I'd like to know, are there certain odor receptors in humans which could cause stress? There's nothing known about that. It's completely unexplored.
We'll go back to the room. There are a few questions up there.
First, thank you for the very nice talk. During the human pregnancy, the sense of smell gets more sensitive. So it's relatively easy to hypothesize why is it, because it has a protective function. What is known about it?
Well, I don't know anything about it. Of course, I've heard about this my entire life, and pregnant women wanting to eat dill pickles and ice cream, but I don't think there's any known explanation for it. I would imagine it has something to do with the hormonal changes affecting the brain. There are hormone receptors in the brain, and hormones can affect neuronal function.
It also turns out in pregnant women, their microbiome changes quite a bit, both their vaginal and their gut microbiome. So that is all the bacteria that are inside of us. In pregnant women, they change quite a bit during pregnancy. And we know some of that probably affects behavior.
Il y avait une autre question juste derrière.
Yes, is there any difference between the perception of a female and male mice or even in humans in terms of odor?
It's been claimed that female humans may have a more sensitive sense of smell, but I don't think there's clear evidence for that. I mean, certainly in mice, in terms of instinctive behaviors, you can see that pheromones will cause different behaviors in males and females.
But you don't think we select mates based on smell, huh?
Well, there's been a lot in the press about that, pheromones and women recognizing their own husband's T-shirt and things like that. It's not well established.
There was this hypothesis, right, that women living under the same roof for quite a long time get their periods synchronized, like sportswomen or nuns or so. Mm-hmm. How important is that study or that finding?
Well, that was done with college students that were living in the same dorm room. And although that's been around for decades, I don't know if people have tried to repeat it and failed or what's happened, but that's still like the only instance by this one researcher that's shown that. So I'm not sure exactly where that stands.
But do you believe there are human pheromones, right?
I don't know. There could be.
By the way, I do know mosquitoes are attracted to different people, right? You know that. And some of this is carbon dioxide. That's well sensed from these receptors. But actually, there's been correlations between the bacteria on people's skin and their attractiveness to mosquitoes. So probably through the pheromones or the compounds these bacteria make. And so it is a real fact that people are attracted differently, and it depends on the bacteria that are on your skin. So you can think about that when you go outside next time.
Well, the question is does that have an effect on other humans?
People's behavior? Yeah, I don't know, but it certainly affects the attractiveness to other animals anyway. Yeah. In this case, mosquitoes.
There's a question here in the front.
I have a microphone here.
Is there a mic coming here? Oh, sorry.
I might have a question. Yes, thank you.
I can't see you. Okay.
If you understood correctly, you did mention that human beings can perceive differently odors when there are different concentrations, if I understood correctly. Do you have any examples, and can you explain that, please?
I just know of one example that an odor chemist told me, and it's called thioterpineal. And at one concentration, it smells like grapefruit. At another, it smells like tropical fruit. And at a high concentration, it smells putrid. But of course, as you raise the concentration, you're recruiting additional receptors, right? So at different concentrations, you have different combinations of receptors.
Thank you.
There's a question in the front here.
How come we get used to a smell so much that we don't even smell it anymore?
Oh, that's a very interesting question. What happens is that the neurons in the nose, if they're continuously exposed to an odorant, they stop responding. So they habituate.
Est-ce que je peux vous demander, on va retourner vers Twitter avec quelques questions. Okay, in the meantime, I'm having quite different questions, right? Everything now seems to be settled by the genes, right? Including diseases. And we all know those firms on the internet that offer to screen your genome for $1,000 or even less now to make a diagnosis in a way for your diseases. But at the same time, the doctors are not trained to this emerging and huge field, right? So will that be an issue for doctors to be trained in that, or will we have to go to kind of genetic doctor counseling in the future?
It is a serious problem. We'll probably talk about that a little more tomorrow, but yeah, I don't think doctors are at all trained for this. So we'll have to change our medical education, because it happens now in California. People will show up with their genetic tests, and doctors, they're clueless as to what to do with this. It's also true that many doctors don't realize for some diseases like cancer, it actually is incredibly beneficial to get your DNA sequenced. And not all doctors realize that, and it's very valuable for certain kinds of cancers. So we really do have to keep physicians up to date in this rapidly emerging field.
Un micro vers...
Oui.
Ah, pardon.
Well, there is a question about what can the industry, probably, I guess, the fragrance or the food industry, take from your work about the receptors, about the processing of information. What do you think is could be of interest for these industries? I guess there are some people from the industry in the audience.
I think eventually, if we could have more information about what the different receptors recognize in terms of the odorants, I've always liked the idea of being able to express all of them in individual cells that you could grow in tissue culture, and then just exposing them to Chanel number 5 and seeing which ones were activated. And just to see, like, to understand how many are activated by a particular odorant. Can you block? Do some block other ones?
I'd love to see us match cheeses and wine with our receptors. I really do think at some level that will come, because people do have different receptors in their bodies. And so I think at some level, once we get the mat made, you will be able to do that at some level.
Qui a encore des questions?
So a question about the sensory system. So what you described is at least work in mice and in mammals, in fact. What about other life forms that may not share this type of receptors? Maybe insects, for example.
Well, as you know, insects also detect odorants, and they use the odorant receptor family that I described is evolutionarily conserved from teleost fish up to humans, so in invertebrate animals. But insects have completely different receptors. Like those in mammals, they're expressed on neurons that send signals to the brain of the animal. And they're quite important to the insect in terms of finding food and finding mates. I think without those odorant receptors, they probably would not survive.
Il nous reste quelques minutes pour une ou deux questions. Alors, une devant et une derrière, peut-être.
Yeah.
Il a déjà le micro.
You haven't mentioned the connection between the sense of smell and age. So is the sensitivity better when you're young, or does it deteriorate when you're old?
Well, like most things, it goes downhill. It's thought, not in everyone, but there's a general trend in that direction with age.
Est-ce qu'il y a encore une ou... Alors, une question en haut pour madame et peut-être en attendant de ce côté-là. Alors, il y a monsieur qui veut... Est-ce que vous arrivez à lui donner un micro ici, s'il vous plaît? Devant la caméra.
Sinon, je peux poser ma question.
Oui, s'il vous plaît.
So I have a question regarding the development. You nicely showed that these ORs are sort of in the immature network, not yet so focused. And we recently did some olfactory studies on functional MRI with newborns only 2 days old. And they gave a very nice response to actually non-biologically relevant new odors that were synthetic. But they don't differentiate them in their cerebral response. Do you think this has to do with yet to be developed receptor specification, or do you think we just didn't pick it up?
What were you measuring?
We're measuring the olfactory response from the olfactory bulb to the piriform cortex into more complex networks like the orbitofrontal cortex in response to biologically not relevant synthetic odors in newborns.
Wow, I really don't. I can't comment. Because you're looking at the whole brain, right?
Yes.
Yeah, so I mean the brain
But there was no differentiation between the response. They responded to all synthetic others in a similar way.
Right. But the nose could be distinguishing, but the brain not.
That's a possibility.
So it's hard to know.
I was interested to see that you ORs in the immature brains distributed much more receptors, right?
Yeah, that could be part of it. But the brain keeps developing even into the teens and early 20s, right?
Oh, I was just going to say this would be great for Romulus and Remus and also Tarzan movies of the future. So knowing that your environment could affect your olfactory receptors.
A question here.
Yeah. Let's suppose that you know all the antagonists for one molecules. Do you think that the smell will be blank, or are we going to smell anyway something?
If you gave an antagonist for all of the odorant receptors?
For one specific molecule, for example. Is it going to smell or not?
Sorry. If you had a...
If you know all the receptors for one specific molecules, and then you screen and you find out all the antagonists, do you think that the results will be a blank odor, or it must smell anyway?
I'm not sure. I mean, I think that, like I said, you probably have different odorants. Some can act. We know that a given receptor can be activated by one odorant and blocked by another. And so it's going to be a very complex interplay between the different odorants and the different receptors, the concentration, etc.
Est-ce qu'il y a une dernière question dans la salle? Ou sur... voilà. La dame là-bas. En attendant, Yvan, est-ce qu'il y a une question sur Twitter?
Les questions déjà soulevées.
D'accord.
Yes, I have a question on Alzheimer's disease. As you know, the olfactory dysfunction is one of the most documented first sign of Alzheimer's disease in humans. So you were talking about neurodegenerative diseases, and I was wondering, from both of you, can we think that olfactory dysfunction could be a biomarker of disease, predictive disease? And is that more on the odor receptor or in the expression of the gene that this would happen?
Well, I think that's actually been known for many years, that it's an early sign of Alzheimer's, and now it's in the news again. I think there are more studies, but I think that the brain is just not functioning. It doesn't, I've always been interested in the possibility that the bacteria or viruses are getting into the brain through the nose and causing disease, but I think that it's true that it's an early marker, but it's not clear why, and there's no evidence that right now it's happening in the bulb or in the nose, for example.
So we don't know the process?
Well, it's not. There are other areas of the brain where you do see damage early on. It's not in the olfactory bulb. For example, it's in the entorhinal cortex and then the hippocampus.
Yeah, I was just going to say, consistent with that, some people are working on early tests for Alzheimer's that are eye tracking tests that seem to appear very early as well. So it's probably more generalized function than just olfactory receptor.
That's interesting. That's probably easier to test also than olfactory abilities.
But do you study Alzheimer mice?
No.
Voilà, est-ce qu'il y a une toute dernière question dans la salle? Oui, et ce sera la dernière. Anosmia.
Oh, what about anosmia?
Mm-hmm.
Well, there's general anosmia, which is where a person cannot smell anything, but that's more unusual. Then there are specific anosmias where a person cannot smell a particular odorant at a low concentration.
Like you, right?
That, yes, I'm a nosmic, unfortunately for musk, which really upset me when I first learned it because I like perfumes. So, but about 12% of people cannot smell musk. In fact, I can't even smell it at a high concentration.
So have you had your repertoire sequenced?
No, I haven't. But there's another one called androstenone, which is a pig pheromone, and about half of people cannot smell that. And there was a receptor identified for that will account for some of the genetic variability.
Voilà, je crois qu'on arrive au terme de cette première soirée. Je remercie nos deux panélistes, surtout Linda Buck pour sa présentation.
Thank you.
Cette discussion va évidemment se poursuivre ces 3 prochains soirs sur ces 4 prochains jours. Demain, c'est donc Michael Snyder qui sera présent ici. Je vous encourage évidemment tous et toutes à venir écouter sa présentation. Si vous avez aimé celle de Linda Buck, la Fondation Wright organise un petit sondage pour voir qu'est-ce qu'on peut améliorer pour les prochaines fois. Donc, n'hésitez pas à le remplir. L'adresse est mentionnée ici, c'est tout simple, colloque.ch/sondage. En conclusion, il me reste à mon tour à remercier la Fondation Wright pour l'organisation de cette soirée, l'Université de Genève pour héberger les Colloque Wright et bien sûr tous ceux qui ont participé à l'organisation de cette soirée et de tout le colloque de manière générale. Je rappelle aussi le spectacle, ça a été mentionné avant, au Bastion, tous les soirs, à différents horaires que je ne connais pas par cœur, mais qui se trouvent sur le site. Et voilà, il me reste juste à vous souhaiter une bonne rentrée et une bonne soirée à tous. Merci beaucoup. Thank you very much. Thank you.